Plants are factories that manufacture yield from light and carbon dioxide. But parts of this complex process called photosynthesis are hindered by a lack of raw materials and machinery. To optimize production, scientists from the University of Essex have resolved two major photosynthetic bottlenecks to boost plant productivity by 27% in real-world field conditions, according to a new study published in the journal Nature Plants. This is the third breakthrough for the research project named RIPE, that is, realizing increased photosynthetic efficiency. However, this photosynthetic hack has also been shown to conserve water. Like a factory line, plants are only as fast as their slowest machines. The researchers have identified some steps that are slower, and what they are doing is enabling these plants to build more machines to speed up these slower steps in photosynthesis. The RIPE project is an international effort led by the University of Illinois to develop more productive crops by improving photosynthesis, the natural sunlight-powered process that all plants use to fix carbon dioxide into sugars that fuel growth, development, and ultimately yield. A factory's productivity decreases when supplies, transportation channels, and reliable machinery are limited. To find out what limits photosynthesis, researchers have modeled each of the 170 steps of this process to identify how plants could manufacture sugars more efficiently. In this study, the team increased crop growth by 27% by resolving two constraints. One in the first part of the photosynthesis, where plants transform light energy into chemical energy, and one in the second part, where carbon dioxide is fixed into sugars. Inside two photosystems, sunlight is captured and turned into chemical energy that can be used for other processes in photosynthesis. A transport protein called plastocyanin moves electrons into the photosystem to fuel this process. But plastocyanin has a high affinity for its acceptor protein in the photosystem, so it hangs around, failing to shuttle electrons back and forth efficiently. The team addressed this first bottleneck by helping plastocyanin share the load with the addition of cytochrome C6, a more efficient transport protein that has a similar function in algae. Plastocyanin requires copper and cytochrome requires iron to function. Depending on the availability of these nutrients, algae can choose between these two transport proteins. At the same time, the team has improved a photosynthetic bottleneck in the Calvin-Benson cycle, wherein carbon dioxide is fixed into sugars by bulking up the amount of a key enzyme called a SBPase, borrowing the additional cellular machinery from another plant species and cyanobacteria. By adding cellular forklifts to shuttle electrons into the photosystems and cellular machinery for the Calvin cycle, the team also improved the crop's water use efficiency, or the ratio of biomass produced to water lost by the plant. In their field trials, the researchers discovered that these plants are using less water to make more biomass. The mechanism responsible for this additional improvement is not yet clear, but the researchers are continuing to explore this to help understand why and how this works. These two improvements, when combined, have been shown to increase crop productivity by 52% in the greenhouse. More importantly, this study showed up to a 27% increase in crop growth in field trials, which is the true test of any crop improvement, demonstrating that these photosynthetic hacks can boost crop production in real-world growing conditions. RIPE's first discovery, published in the journal Science, helped plants adapt to changing light conditions to increase yields by as much as 20%. The project's second breakthrough, also published in Science, created a shortcut in how plants deal with a glitch in photosynthesis to boost productivity by 20 to 40%. Next, the team plans to translate these discoveries from tobacco, a model crop used in this study as a test bed for genetic improvements because it is easy to engineer, grow and test, to stable food crops such as cowpea, maize, soybean and rice that are needed to feed our growing population this century. The RIPE project and its sponsors are committed to ensuring global access and making the project's technologies available to the farmers who need them the most.